Happy Friday, everybody. This video is about mitosis, and the slides are set up so that you can fill in the blanks on the third page of the note sheets that I gave you for the last lesson. So the last one was on cell cycle, and this one is the third page, which is on mitosis. However, if you don't like the note sheets, or if you're finding it awkward to watch videos and take notes at the same time, just given the small size of your iPad, you are welcome to handwrite this. You're welcome to just draw out the steps, whatever it is that works for you. All right, so let's just review from last time. We were looking at the cell cycle. And we learned that all cells start out in G1 or gap 1, where the cell is getting bigger, it's growing, it's doing cell things. Then it moves on to S phase, or synthesis, where the DNA is duplicated. And then it moves on to G2 or gap 2, where it prepares to divide, copies the organelles, and gets set for mitosis, or M phase. So we're at M phase right now. Before we do that, we actually have to spend a pretty good amount of time talking about chromosome vocabulary, because it can be very, very confusing. We will repeat a bit of this when we do meiosis after spring break. But for the time being, and this is in your notes, eukaryotic chromosomes consist of chromatin, a complex of DNA and protein. We talked a little bit about that when we talked about eukaryotic regulation. So chromatin is basically your DNA kind of all tangled up around those proteins. Every eukaryotic species has a different number of chromosomes, and you might remember you've seen these two terms, somatic cells and germ cells. So somatic cells are going to be cells that have 2 sets of chromosomes, and germ cells, or gametes, have 1 set of chromosomes. So chromatin overall, just this mass of DNA with protein, and then how many chromosomes does each organism have kind of depends on the species. And then somatic cells having 2 sets, and then germ cells having 1 set. So what do we mean when we say that we have 2 sets of a chromosome? Well, in a somatic cell, that would look like this. So this is the first kind of chromosome that a human has, chromosome number 1. And here I have 2 sets. I've got this one, which came from this person's mother, and this one, which came from this person's father. 2 sets of the same kind of chromosome that have the same kind of genes in the same spot. So if this is a gene for eye color, this would be another gene for eye color. And then here's, let's say, the 15th chromosome. This one came from this person's mom, this one came from this person's dad. And let's say here, if there is a gene for making testosterone, here is also a gene for making testosterone in the same spot. 2 sets of chromosomes. Normal body cells, somatic cells, have 1 of each kind of chromosome, 1 from each parent. The chromosomes in a germ cell, or a gamete, so this would be a sperm or an egg, would come in a set of 1. So 1 of chromosome 1, 1 of chromosome 2, 1 of chromosome 3. So you might be wondering, well, where did these come from? Are they this person's mom or this person's dad's chromosomes? We'll get to that later on. That's meiosis, but the short answer is it's random. So this one might have come from his mom or his dad. This one might have come from his mom or his dad. If you're confused, the big point here simply is, and sperm cells or gametes or eggs only have 1 of each kind of chromosome. And just briefly, that makes sense, right? Because you would have, let's say, an egg and a sperm, 1 set of chromosomes in each one, combined together to make a cell with 2 sets of each chromosome. In a really simple fashion like this. 1 of each kind. Comes together to make 2 of each kind. Alright, so that's chromosomes. So how are these chromosomes kind of distributed, and what do we call them? So in preparation for division, the DNA gets copied. Remember, that's an S phase. And you know how that happens, where the DNA is unzipped with helicase and untwisted with topoisomerase, etc., etc. So then each duplicated chromosome has 2 sister chromatids, which are joined identical copies of the original chromosome. The centromere is the spot where the two chromatids are most closely attached. So it's like a little... So this is a great real photograph of a chromosome that's been duplicated. I've got two sister chromatids, one here and one here, and they have identical DNA. So if over here I have a gene, let's say this is a plant and this is a gene for making pink flowers, over here I would also have a gene for making pink flowers. I can't have one gene for pink over here and one gene for white over here. That doesn't make sense because this is simply identically replicated DNA. So remember when we did DNA replication, and we have like, this is like the original DNA, and then I would make my new DNA like that. Sorry for the drawing. And so this is this side and this is this side, just those two identical copies. I mean, yeah, that's the main idea. So you might call this whole thing one chromosome with two separate chromatids. And then when they're separated, then I have... Alright, so during cell division or mitosis, the two sister chromatids of each duplicated chromosome are going to separate and move into two separate nuclei. Once separated, the chromatids are now called chromosomes. Two chromosomes. So once again, this is one chromosome that is unduplicated. I haven't copied it yet. We're still, let's say, in G1 or gap1. Over here, I also have one chromosome, but it's duplicated, and it's got two sister chromatids. This used to be very, very confusing to me. I hope it's not to you. So one more time, here would be my cell, let's say in gap 1, and then I have copied it in S phase, and now I have two identical sister chromatids. And then, so this was like S phase, and now I do mitosis, separate those two sister chromatids into two separate cells. So here's my one chromosome, which has now become two separate chromosomes. So one more concept here for chromosome vocabulary is homologous chromosomes. In humans, we have two of each kind of chromosome, as we talked about earlier, and one of the chromosomes is maternal from the mother, one is paternal from the father, and they have the same kinds of... I really like this picture. It's worth pausing and reading the text, but I just want to focus on the picture for now. So I have a set of homologous chromosomes here, and the homologous chromosomes have already been duplicated. So over here, I have, let's say, the chromosome that came from this plant's mother, so it's the maternal chromosome. And over here, I have the paternal chromosomes. And each of those chromosomes has two sister chromatids, 1 and 2. And the colors represent the fact that the mother's chromosomes here that are duplicated have two identical genes. So let's talk about trichromes. Trichromes, which are little hairy things that plants will have that protect them from predators. Over here, let's say I have the gene that makes trichromes, so let's put a plus for that. And I have the exact same gene, the one that makes it and the one that makes it, on these two sister chromatids because they're identical. Whereas over on the paternal side, it has a different color, and that's kind of representing the fact that I might have a different version of that. Do you remember those are called alleles? So I still have the gene that is concerned with making trichomes or not, but maybe over here I don't have that version. Maybe I have a different version that doesn't make them, or that makes them taller or smaller or something like that. So they've got these, here as it says, these regions that code for the same gene. They just might be different versions of the gene, or they could be identical. This could be the gene that does make trichomes, but the point is they don't have to be. Right, so those are homologous chromosome pairs, one from each parent. They get that this is a homologous pair. But this can be very, very confusing for kids. I just have a variety of different pictures here to help show this to you. So once again, homologous pair, one from dad, one from mom. Sometimes kids think, like, they get that this is a homologous pair. They don't understand that both this and this represent homologous pairs. It's just that here, I haven't copied the DNA yet. Here in S phase, I've copied them, and now they look like this. But in both instances, they're both homologous pairs. Last picture, I think here, we've got, for homologous chromosomes, we've got a karyotype. A karyotype is a picture of all the chromosomes for a human being. So this is showing a homologous set, all the chromosomes. Purple, let's say, representing the ones that are maternal, and blue representing the ones that are paternal. And I have one of each kind. 23 homologous sets. Here I still have 23 homologous sets. It's just that we've gone through S phase, we've gone through replication, we haven't divided yet, and now I just have duplicates of all of those. Hopefully that's making sense. All right, so just a quick point. I mentioned earlier that every organism has a different number of chromosomes. So for example, mice actually have 20 pairs of chromosomes. And you can see that right here. 1, 2, 3, all the way up to 20. And this is showing their homologous sets. One from mousy mom, one from mousy dad. And there's actually a mistake in here. If you could take a second to see if you can find that. It's right here. It actually has an extra 17th chromosome. We'll talk about that when we get to meiosis. There is a word that's used here called diploid. I will come back to that when we do meiosis because it's confusing. And puppies have their own sets of chromosomes. They actually have 39. I like dogs, and no offense, Sam, but they're not necessarily more intelligent than humans, although some might argue about that. But so the fact that they have 39 chromosomes should tell us that the number of chromosomes isn't really related to the complexity or the intelligence of an organism. All right, let's get into mitosis then. These slides just have some things for the College Board. If you're concerned about what you have to be able to do on the AP test, you can read those. All right, so getting into it, this is where your notes start. So eukaryotic cell division consists of mitosis and cytokinesis. So mitosis technically is the division of the genetic material, So that means it's where the DNA is actually splitting. And then cytokinesis is where the cytoplasm is actually going to split and the cell pinches off into two. Remember that gametes or sex cells are produced with meiosis. And meiosis is going to give us 4 non-identical daughter cells. So all of the different sperm and egg produced in meiosis are going to be different from one another. And they will result in one set of chromosomes, half as many as the parent cell. So again, we want our sperm and egg to have only one set of chromosomes. They should be missing their homologous partner so that 23 chromosomes in one gamete plus 23 in another gamete give me a set of 46 chromosomes, at least in humans. So that's the process of meiosis, and we're not doing that today. I just wanted to talk about the difference. Again, in meiosis, we have 4 cells, and they're not identical. Mitosis, on the other hand, is not going to give us 4 chromosomes, or 4 cells. It gives us just 2, and they're identical. So in mitosis, we have 2 cells, and they are identical. Basically, how mitosis works is the chromosomes, which have been replicated in S phase, line up in the middle of the cell, they pull apart and make 2. This is one of the best videos about cell cycle, and it also reviews mitosis at the end, so we'll watch this. I'll also put it in my link so that you can watch it again later if you need to.
In cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into three distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
All right, hopefully that was helpful. So, what we'll do next, now that you've gotten a chance to see those things in action, is look at some pictures and talk about what's happening. So in your notes, basically all you have to do right now is write down the different names of the steps. So there's blanks there, and these are in order. So first one, prophase, then prometaphase, metaphase, anaphase, telophase, and cytokinesis. So let's start going through steps. So this is what the cell would look like inside of interphase, G2, where I'm preparing to divide. You might remember these terms centrosomes and centrioles from when we talked about the different cell organelles in our, what do we call it, the speed dating activity that we did. So centrosomes are just this body, remember the word some means body, this body of fibers, and specifically centrioles are the really tightly packed sets of proteins or tubules that are going to be moving the chromosomes for us. Check out your DNA. Here it's all just one big ball of chromatin. They're not condensed. And then moving into prophase, I start to have my centrosomes start to separate. The tubule fibers or the mitotic spindle starts to form, and my chromosomes are all condensed and ready to go. And I have two sister chromatids, and we can easily visibly see them here. I did have two sister chromatids here as well. They were already duplicated, of course, because that happened in S phase before G2. But now here I can see them. So here it was like all in one, this kind of white part here and here as well. Moving into prometaphase, which by the way was a phase that did not exist when I was in high school. They just started kind of naming it shortly around that time. In prometaphase, the nucleus starts to break apart. So here's my nucleus, and it's now kind of in pieces. And here it started to break apart. And if you think about it, that makes sense. I'm trying to pull the chromosomes apart into separate cells, and I can't do that if there's a nucleus in the way. In prometaphase, they're also kind of moving towards this process of lining up. So my tubules, or my mitotic spindle is starting to form and starting to attach to my chromosomes. In metaphase, they line up in the middle, and I always remember metaphase because M for middle. And then in anaphase, which I remember because to me, this looks like an A. So this is anaphase. In anaphase, I am pulling apart the sister chromatids. So these little fibers are just basically contracting and pulling them towards the poles or the ends of the cell. In telophase, the nuclear envelope starts to reform, the chromosomes are decondensing, and in cytokinesis, I am pinching off the cell membrane to form two different cells. So in pictures, and this is actually a plant cell, this is what it would look like. Here's prophase, chromosomes are condensing, starting to form the mitotic spindle. In prometaphase, the nuclear envelope is breaking down. Metaphase, they're lined up in the middle. Anaphase, they're starting to pull apart. telophase, my nucleus is starting to reform, chromosomes are decondensing, and I'm starting to form, I'm starting to have cytokinesis happen. There's a few other pictures here. If you don't like the other ones I just went through, these are quite good. Garbium and pause them. And then let's talk specifically about cytokinesis. So in animal cells, cytokinesis occurs by a process known as cleavage, forming a cleavage furrow. And in plant cells, we get what's called a cell plate. So this is the cleavage furrow, just a little pinching off of the cytoplasm. And there's actually a little contraction happening there with a special kind of fiber that I'll show you in a second. I just think this looks really cool. And cytokinesis in a plant happens a little bit differently. It forms this thing called a cell plate. So it has these little vesicles that pop up. They kind of fuse together and form this whole new cell wall and cell membrane. Remember, plants have a cell wall and a cell membrane. Animal cells just have a cell membrane. So that's how cytokinesis looks different in animal cells versus plant cells. So I really like this video. It shows real cells doing mitosis. These are nematode cells, which are a type of worm. And what they've done is used fluorescent proteins to mark the different molecules. So they took a jellyfish gene, and using transformation, which you know how to do because you did it in Madison, they inserted the gene into the nematode's DNA. And they used a green glowing gene to mark the microtubules, which are the things that pull apart the chromosomes, and they used a red glowing gene to mark myosin, which is a different kind of protein. If you took anatomy, you've probably heard of that before. So let's see what happens. So in the beginning here, just kind of focus on the cell. We have prometaphase happening. So these are the two nuclei, and they're about to divide. I'm going to pause this really quick. You might see these kind of dark spots. This is where the nucleus is. And down here, these are a little bit farther along. Those dark spots are where the DNA is. And the reason why we can't see them is because they don't have myosin or actin in them. So they're kind of like these black holes, they call them. All right, so now we're going to start getting into metaphase because we've broken apart or about to break apart the nucleus. Here we go. There's the nucleus breaking apart. And there's metaphase happening, and now anaphase is going to happen. We're going to pull. The mitotic spindle is pulling them apart. There they go. Pulling. They're pulling. Isn't this exciting, guys? There's my centrioles, or my centrosomes, full of those fibers. There's a really cool lab you can do in college cell bio where you get to take pictures of cells doing this and talk about how to change it. Okay, so now we're starting to have the mitotic spindle disassociate, which means we're starting to get into telophase. Well, maybe not quite yet. There's my chromosomes still undergoing anaphase. There's the cleavage furrow starting to form, and then it's done. So that last phase was telophase and cytokinesis happening. And you can see the myosin starting to take place here. It's trying to kind of reform those cells and put them back together. All right, that was exciting. So just some last comments. All cells have to be produced using some sort of cell division. And what we just saw in mitosis is called binary fission in prokaryotes or bacteria, and basically works in a similar way. Here's my original cell. Here's my original cell. Copied the DNA in S phase. They get pulled apart. We split them down the middle in cytokinesis, and then to them we have two different cells. Your instructors in Madison used this term binary fission, but you didn't know it yet. So mitosis in prokaryotes or bacteria is just called binary fission. Since prokaryotes evolved before eukaryotes, mitosis probably evolved from binary fission, which is kind of cool. So then just some quick practice really fast here. So how many chromosomes are in the middle cell? Pause it and think about it. If you said 1, you are correct, because remember that when you have a sister chromatids that are duplicated here, we still call this one chromosome. even though there is 2 identical copies, generally refer to this as 1. Sometimes I find it helpful to think of it as 2, and we'll talk about that when we get to meiosis, but technically most people would say this is 1 duplicated chromosome. At what part of the cell cycle would you see a chromosome that looks like this? Hopefully you were thinking it would be after S, so we have to already be into G2. Okay, I can't be in M phase yet, or mitosis, because I haven't started to divide. And I can't be in G1 because I haven't duplicated the DNA yet. I could be, finished with S, but the best answer is G2, because I've copied the DNA and I'm all ready to divide. Although the chromosome began M phase, looking as shown below, when do these sister chromatids separate? So the question is, when what part of mitosis do I actually pull apart these sister chromatids? And hopefully you're thinking anaphase. Remember, anaphase is when they pull apart. If you were to examine a sample of connective tissue cells from a small but adult mammal and a second sample from a fetus of the same species, in which would you expect to find more cells undergoing mitosis? So would it be the adult or the fetus? Pause and think about that. Hopefully you've decided the fetus, because they are growing quickly, therefore they're going to have mitosis happening more in their cells. All right, I know that was a lot. Hopefully you're feeling relatively good about this. Like I said, drawing some pictures of this would probably be to the benefit of many of you. And we'll talk today in WebEx or throughout the day if you have any questions. Bye.